we're going to look at the second component of the ecology unit, which is population ecology. And population ecology focuses on factors affecting population size over time. And so what we're really looking at is two different things. We're looking at density and we're also looking at dispersion. And population density refers to how many individuals are in an area or a certain unit area or a volume in a given area, and dispersion looks at the pattern of spacing among individuals within the boundaries of the population. This picture shows an iguana, several iguanas actually if you look closely, that live in trees over a river in the northeast corner of Costa Rica. And these iguanas, which are cold-blooded, meaning they don't generate their own heat, but they've adapted to live in a wide range of environments. And while they're usually found near water or rainforest, as is the case with the iguanas found here, they can also be found in the desert and in other environments where they get their water from their food sources instead of getting their water from a river, or lake, or a pond. So when we look at distribution patterns, we have three basic types. We have a uniform distribution, we have random distribution, and we also have clumped. And so when we talk about population dispersion, we're really talking about how often they occur in this area, and then we're looking at these three different types. So uniform dispersion results from intense competition or antagonistic behavior between individuals. And they're relatively rare, and they only occur where environmental conditions are fairly uniform. And that's why you have this intense competition and antagonistic behavior between these organisms, because there isn't going to be much changing. The second distribution pattern is random, and this occurs where there is no competition and no antagonism or a tendency to aggregate, and the conditions are uniform. It is rare for these conditions in the environment to be met, so it's very rare that you're going to see any kind of random distribution. So the second type of distribution pattern we have is random distribution, and this occurs where there's no competition, no tendency to aggregate, and no antagonistic behavior. The conditions are also uniform, and it's rare that all the conditions in the environment are met. The third type of distribution pattern is clumping, which is the most common distribution pattern. They're seldom uniform. The reproductive patterns favor clumping, and animal behavior patterns often lead to congregations. So, meaning by animal behaviors, it's beneficial for them to clump together in a group. The optimum density for population growth and survival is often an intermediate one. Undercrowding can be as harmful as overcrowding. So there's this optimal clumping number in different populations that makes it the safest for whatever reason or the most beneficial for that group. So having too much or too little in a clumping distribution can be a problem. So what I would ask my students here is to give me some examples of specific clumping distributions that they've seen. And here I have some cattle, some geese. I'm also showing a flock of fish that stay together, as well as some plants that stay together in nature. And so why would it be beneficial for organisms to be clumped together? One of the reasons is to protect or alarm, to sound off an alarm if there's a predator. The other one is to conserve energy resources, so they do different jobs within their group. There's a lot of different reasons why they would want to clump together. So population dispersal. Natural range expansion shows the influence of dispersal on distribution. And so what it means by that is that there's a natural range sometimes that explains why organisms live where they live. Cattle egrets arrived in the Americas in the late 1800s and have expanded their distribution. And so the reason why that has occurred is because as the cattle have expanded into this area, the egrets come and have a beneficial relationship with them. Population dispersal. There are rare cases where you have this long distance dispersal that can lead to adaptive radiation. And in the picture here, you have the Hawaiian silver swords, and they're a diverse group descended from an ancestral North American tarweed. What's important about this is that adaptive radiation is the diversification of an organism or group of organisms into forms filling different ecological niches. You wouldn't normally find that here, but it's found a niche here and it's able to survive. And so it's really important that you think back about adaptive radiation. Some other examples of this would be Darwin's finches, marsupials in Australia. If they can find a niche that meets their needs, then you have this long distance dispersal for whatever reason, whether it was generated by accident or on purpose. So let's take a look at the African honeybee and when it first came to America and how that has happened. So I've shown you a map here in South America where the honeybee was first brought over back in the 50s. And it was brought over because it was very popular with Brazilians in terms of raising honey because of their behaviors. And so what happened is that the researchers brought this bee over to Brasilia to improve this productivity because the Brazilian bees weren't cutting it. And so how long did it take once this came in the mid-50s for it to reach the United States on its own, getting out of the hive and all of that? It took 40 years. So we have a large wild population that quickly developed and spread through South America, Central America, and Mexico. And in the 1990s, this honeybee was identified in Texas and it has since spread throughout the Southwest. It's successful, and I could ask you why it might be successful, and there are a lot of correct answers here. One, it produces more offspring. They tend to defend their nests more fiercely and in greater numbers, and they're often likely to abandon the nest when threatened by adverse environmental conditions or predators. So they have a lot of adaptation ability, and as you can see, it took right around 40 years for it to reach the United States, and they've actually had sightings of this bee as far north as Tennessee. So some organisms have small geographic ranges, while others have large geographic ranges. And so I'm just looking here at a bird, a North American bird and a British vascular plant, as illustrations that they don't go far from where they're normally found. Species with large geographic ranges, it could be a whole bunch of different areas, but generally you find things in coastal areas that are going to move and have the ability to move to other areas and find suitable niches for themselves. But it's very rare that you find species that have a large geographic range. But again, as you look at what's going on here worldwide, continentally and regionally, you're really finding a lot of these by areas of water where the water's doing the moving, or maybe you have a high shipping trade that's able, that makes it beneficial. The species has a methodology of being able to move into other areas. So here's a slide that shows the interactions between organisms and their environment, and this determines how abundant that organism is. And so I would ask my students, why do you think the range of kangaroos followed this pattern? And so what we have here is this is the continent of Australia, and climate in northern Australia is hot and wet with seasonal drought, and these red kangaroos occur most densely where you see this dark brown, and they like it where it's arid or semi-arid, precipitation is relatively low and variable from year to year. And as you can see, it has these pockets here where they're pretty numerous, the red and the dark brown. But then it has some orange pockets where there's some numerous adaptations for the marsupials there. But there would be lots of reasons for that. So how do we know how big a population is and why do we care? So there's three basic ways that you would estimate population size. The first one being that you would count it. How many do you have? If it's a small enough population, you can just simply count it. The second way is to divide the area into quadrants and count the number of individuals in several quadrants and then estimate the entire area. So quadrant sampling, you use the random sampling method to determine what quadrants you're going to count in, and then you multiply that by the number of total quadrants to try to determine how many you have. And then the third way, down here in the bottom where you're looking at the crab, is mark and recapture. And you take a limited number of individuals, you capture them at random, you mark them, and you can, this has been called tagging. Sometimes they put little clips or homing devices in them. It just really depends on how much money you want to spend on the tagging system, and or die or marks. And then a later time, a second group of animals is captured at random from the population, and the percentage of marked individuals determined. And so then you're able to do this calculation. If 10% of the animals in this second group is recaptured, then the original 20 represents 10% of the population. So the total population would be 200. And so we captured 20, we marked them. In the second group that we picked up, 10% of the animals in the second group were already marked from our first marking. And so now what we say, because of that we have 10%, then we know that the population is 200. So we do a lot of mark and recapture techniques as well as quadrant sampling. And where this comes into play, and I'm going to go over to the calculation part in just a second, is if you live in states where they allow hunting, where they issue fishing licenses, how they figure out how they're going to keep the population from overreaching carrying capacity is they do a population sampling to determine the number of organisms that they want to eradicate or get rid of. And so some years in Virginia, we get what you call tags when you are a deer hunter. And some years you get 3 tags, meaning you can kill 3 deers. And some years you can get 6 or 7 tags, meaning that the population has had a boom for some reason, and you can get more tags for that. Same thing with fishing. So it's a way that they estimate the population size to allow these other recreational things to occur that help keep the population in check. So, an emphasis about this method comes with a whopping assumption, and that is marked individuals have the same probability of being captured as unmarked individuals. And so this is a simple formula for estimating population size. And you rearrange it to get the estimated population would be the number of individuals marked and released in the first sample times the total number of individuals in the second sample divided by the numbers of individuals marked and released in the second sample. And so that's how you can determine this population size. Again, you have this assumption that you have the same probability of capturing marked individuals as unmarked individuals. And it's important to understand, especially now in the bio test, that we have more statistics that we're supposed to be working with. It's important for us to understand where there could be some problems or what the assumptions are, because there are a tremendous amount of assumptions in statistics. So let's try an example. 20 individuals are captured at random and marked with a tag. Therefore, the number of animals marked were 20. So we mark our 20 turtles. Let's just say we're going to do turtles. And at a later time, a second group of animals is captured at random from this population. Some will already be marked. Say 10 individuals were marked out of 35 that were captured the second time. So now we know n. So n is equal to 35 and x is equal to 10. And we're going to apply the formula that I showed 2 slides back. So we would multiply the first sample, 20, times the second sample, 35, divided by the x, which was the number of individuals that we found marked in the second group. And that would be 700 divided by 10. And we would end up with an estimate of this population of 70. So if you have questions about any of that, which we just did, you're going to want to go back to this slide here, take a look, make sure you understand what it's asking you, and being able to plug and chug the numbers. And so which method would you use? To determine the number of turkeys in a county, what would be the best method to use to do that? Number 2. So we might use the quadrant sampling. We might use the mark and recapture because turkeys are going to travel. A lot of quadrant sampling is easier with organisms that don't move, like trees or grasses or certain types of fungus and things like that. To determine the number of deer in the state of Virginia, we would probably use mark and recapture. The number of dogs in your neighborhood, you could definitely just go and count those depending on how big your neighborhood is. And to determine the number of feral cats in your neighborhood, this would be a hard one to get your hands on. Feral cats being wild, it would be hard to do that. So you might have to do a mark and like a capture and mark and tag in order to determine that method. So let's take a look at survivorship curves. And so we have type 1, type 2, type 3. And this is basically what do these graphs indicate regarding species survival rate and strategy. So how do you attempt it? So we have a number of species that attack their population strategy is having a tremendous amount of organisms because they know that a lot of them aren't going to survive into adulthood and that they're going to die. So they have a whole bunch. They don't invest a lot of energy in their organisms. And they frequently have organisms because they know this death rate is pretty steep. And then we have type 2, and these are intermediate. They have a constant death rate over the organism's lifespan. And this sort of survivorship curve belongs to like invertebrates, lizards, some annual plants, and like the ground squirrel would be found in type 2. And then type 1 organisms invest a lot of energy. They have low death rates. They invest a lot of energy in their offspring, and they have few offspring, but they do give them good care because they have a higher chance of surviving. Whereas these type 3 organisms, the oyster down at the bottom, is going to release millions of eggs, but many of those organisms are going to die as larvae from predators, and few will survive long enough. Few will survive to the point where they're able to reproduce and have other organisms. So high death rate, constant mortality, very high early mortality, but few survivors, then they live long and stay reproductive. So that's what's important to remember about these survivorship curves. So this is just another curve here showing you the Belding squirrel, humans. If we were in class, I would ask you several questions about these curves to make sure that you understand. They call them life histories now. So let's take a look at this population growth curve. How many more deaths occurred in 2001 than births? So let's take a look at that and see if we can figure out how we figure out birth minus death. So there's a formula at the top. This should also be on your formula sheet. I'm looking to verify that. Yes, it is under population growth, and you are going to see that formula on your formula sheet. So there's no need to memorize this formula up here. It is, however, important that you understand what each of the variables represent. And so in order to figure out birth to death from these years, what we're going to have to do is locate both time intervals. In 2000, the population of perch was 16,000. In 2001, they declined by 1,600 perch. Okay? Therefore, 4,000 more deaths occurred than there were births. Don't let students think this is simple. 4,000 perch didn't die. You need to understand that the births didn't cease. Therefore, okay, let me go back. So let's look at the quantitative analysis of this. So we look at 2000, where we had 16,000 perch, and then we look at 2001, where we had 12,000 perch. So we had a change of 4,000 perch. Therefore, what happened was you had 4,000 more deaths than there were births. So I don't want you to think that this is a simple case of the 4,000 perch dying. That's not the case. And it's not the case that the births stop happening. It just means that the death number exceeded births by 4,000. And so when you put this into a quantitative analysis for the change of the population, 12,000, minus 16,000, you end up with negative 4,000. Therefore, you had 4,000 more deaths than births between 2000 and 2001. And so it's very important that you understand what's occurring here, because a lot of students just think 4,000 perch just died and there were no births, and that's not what happened. So it's really important that you understand when you're looking at a population growth curve, what it's telling us when we have a change. So a change in 1,000 perch from 1999 to 2000 means that they had 1,000 more births than deaths that year. But that's what's important for you to be sure that you understand the difference in. Let's take a look at this. And I'm showing you an exponential growth right next to a logistic growth. And the reason why I'm doing that, and the exponential growth model is in blue. I don't know why it got changed. And this is the idealized population in an unlimited environment. So exponential growth is what would happen if nothing was impacting on this environment. And this can't continue indefinitely. Okay, this doesn't happen. Eventually you reach a carrying capacity for whatever reason and it moves. But we have this exponential model here in blue, and this is showing what it would look like in an idealized population. And we normally refer to this blue as a J-curve. Then you have the logistic model, which is in red, and it considers population density on growth, carrying capacity, which is the maximum population size that an environment can support, and you generally see this with K-selected species, whereas you see the J-curve with R species. So these two formulas are also on your formula sheet, so you are going to need to be familiar with the variables, what they represent, and how you can calculate those on the AP exam. All right. So one bacterium reproducing every 20 minutes could produce enough bacteria to form a layer over the entire surface of the earth, 1 foot deep. But that's only if we're talking about it going unchecked, and that doesn't happen. So one of the most common examples of exponential growth does deal with bacteria, because they can multiply at such a fast rate that when bacteria splits into 2 new cells, it doubles, and then both of them can reproduce. So if we start with one bacteria that can double every hour, since it reproduces asexually, doesn't need to go through meiosis, by the end of one day, we will have over 16 million bacteria. And so it's important to understand, again, the variables here. While the formula is given on the AP exam, variable explanation is not, and you need to be sure that you're familiar with those and comfortable with dealing with them. In the logistic population growth model, the per capita rate of increase, or Rmax, declines as carrying capacity is reached. That's important to note. And the logistic model starts with the exponential model and adds an expression that reproduces per capita rate of increase as n approaches k, which is this carrying capacity. What's also to emphasize here is that logistic, as in logarithm, so be sure you keep that in mind, those of you that are in math class. So let's take a look here. This is new cases of AIDS in the United States. And I just like to put this up as an example. And so if I ask you what is the carrying capacity for AIDS according to this graph, what would you come up with if we were just having a conversation? Where would we come up with? We'd come up with roughly about 45,000 maybe as of 1995, where it's reached carrying capacity. Okay, let's take another look at some more growth curves real quick, just to make sure we've done some good comparisons. So we have this J curve, which is exponential growth, and it characterizes some rebounding population. So an example of that would be the elephant population in South Africa. It grew exponentially after hunting was banned. And so that's really important to note. So we had this really population that wasn't growing very much because we kept having a lot of kills. People would go to Africa to kill these elephants. But as soon as they banned hunting, we had this exponential growth with them. And the logistic model describes how a population grows more slowly as it nears its carrying capacity. And the exponential growth cannot be sustained for long in any given population. Because what happens when they get to this exponential growth is they have either abiotic or biotic factors that impact it. And we have these density dependent factors that impact it, meaning that as the density grows of that population, they start having factors that affect it just because of the population density. The logistic growth curve is usually referred to, produces as an S-shaped curve. meaning sigmoid. And it's a more realistic model because it limits growth by incorporating carrying capacity, and it's the maximum population size the environment can support. This figure shows the population growth predicted by the logistic model. The growth of laboratory populations, such as Paramecium, fit an S-shaped curve. These organisms are grown in constant environment, lacking predators and competitors. So I like this next graphic because it really helps you to look at the difference between the J and the S curve. After the curve is leveled off, births and deaths are in balance and the population has zero population growth. This occurs because the environment is limiting the environmental limitations become increasingly effective at slowing population growth, meaning that there's not enough food, there's not enough space, maybe the organisms have a behavioral ritual that they're not able to now perform because the density is too great. And when the density starts to approach this carrying capacity, the limitation becomes more and more severe. So you have this environmental resistance that becomes more and more severe. A density-dependent limitation, K minus N over K, is one whose effect is determined by the density of the varying population. And so as I mentioned earlier, you have these density independent and density dependent factors, and density dependent factors is at the closer you get to this reaching your carrying capacity, the more the density dependent factors are going to impact this population. Okay. So here is a graph that I would ask you to graph this data given as it relates to a logistic curve. And I want to emphasize that the versus terminology is their friend, since it correctly states as Y versus X or rise versus run. If you're more comfortable with that terminology, you should know this from math class, but this protocol is not always followed among casually created materials. So in this case, the number of individuals is the y-axis and time is the x-axis. So be sure that you're really clear on understanding that, because it's really important that when we put time on the x-axis, that you understand why we're doing that when we're in a situation. And you need to be sure that when you write a graph like this is that you articulate in a paragraph or a sentence why you did it that way. All right, so this is what it would look like. We have this population size, we have this time in years, we have carrying capacity going on up here. And this just shows I just connected the dots. I actually did a line of best connected the dots. You wouldn't want to do that like you did in elementary school. You're really going to properly show that curve. That's really important. All right, so let's talk about our selected species. They're very opportunistic. They have short maturation and lifespan. They're usually pretty small. They don't have a lot of parental care, and they have a high death rate. Compared to K-selected or equilibrial, and this now is returned to like these life histories. They have long maturation and lifespan, few offspring, often large, and usually several late reproductions, extensive parental care, and a low death rate. And it's important to understand the difference between these two because that's just the way they approach making sure their population survives. You have to have a lot of organisms because you know a lot of organisms are going to die. And so here we have a graph of Paramecium as well as a graph of Daphnia. And I would like you to predict on the first one, what is the carrying capacity of the population in this lab? And also, how long does it take the Paramecium to reach K? Whereas when you're looking at the Daphnia, what their carrying capacity is, and how long does it take them to reach K? And so if we were looking at the Paramecium first, they're going to reach carrying capacity somewhere around 900. And it only took 10 days for them to get there. Whereas the Daphnia, their carrying capacity is about 90. Yeah, it's about 90-ish. And it took about 135 days for them to reach that.